Hey everyone, welcome back to the channel. If you're new, make sure to like and subscribe. So in today's video, this is the third part in this series. This is a continuation of the last video. This is microbial cell structure. Now we're focused on the inside of the cell, which is the cytoplasm. So let's start again with this jelly donut analogy. Last time we talked about this, we talked about the outside, the donut, and that would be the cell envelope, right? This time we're talking about the jelly, the cytoplasm, and the stuff within that inner area. So if you look over here on the cell, what you can see is that the cytoplasm is all this right here, all the inside stuff. And so you see there's DNA over there, and just ignore this for now. So let's take a look at what the cytoplasm is. So I keep saying it's like a jelly. It's a gel-like interior of the cell, right? It contains ribosomes, proteins, DNA, and RNA. So that's literally what it is. It's like a gelatinous mixture. That's why I keep saying jelly. Now, just so you remember, DNA is basically the instruction manual of life. And so if you've ever had an instruction manual for any kind of product, it's usually pretty hefty. Now, imagine an instruction manual for your entire existence, right? So that's just to show that there's a ton of it, right? And as you can see over here, this is DNA over here, basically spread out. This isn't spread out all the way, but this is just partially spread out. As you can see, this whole entire thing somehow needs to fit into this tiny 1 micrometer bacterial cell. And so it does do that. And so we need to talk about why or how that works. So what happens is we have a nucleoid. So remember, this is just DNA floating around, right? It has to fit into this tiny cell. And so it gets packaged really tightly. And as you can see over here, that packaging of it is called a nucleoid. So it's not a nucleus because a nucleus has a membrane around it, whereas a nucleoid is just DNA. So let me show you here. This is a nucleoid and this is a nucleus. You have DNA and you have a membrane all the way around. Whereas over here with the nucleoid, there's no membrane. So just make sure you have that down. Now let's take a closer look at the bacterial genome, right? So the DNA of the bacteria. So I've shown you two different views just to show you that it's circular, because this can look a little bit confusing. It is actually just one circular piece of DNA. Now the genome size, so how long it is, it can be from 500,000 base pairs to over 10 million base pairs. And as you can see, that's a very high range. It's a lot. It's a big difference. And so what you can see is that there's a reason for that, right? The reason for that is the ability to tolerate various living conditions will increase the amount of base pairs. So let's think one more time about that instruction manual, right? If you have a cell that needs to tolerate various different environments, it'll need more instructions, right, to be able to do that. And the other thing is more complexity. So if it has a bunch of internal structures, it has appendages, as we talked about in the last video, it has a huge thick cell wall, all these different things, it needs more instructions on how to build it, how to piece it together, how to break it down, how to do basically everything. And so that's just to show you that depending on the bacterial cell that you have, its genome can be from 500,000 to over 10 million base pairs. Now, again, talking about the genome, it's also dynamic, meaning it moves around. And so if we think about DNA, like we said, you have this blob of DNA over here, right? You can't really do anything with DNA. It's sort of your only copy of the instruction manual. So you don't want to write on it and do all these things. So what you do is use transcription machinery. What that does is it'll read the instruction manual and it'll output something that you need to do. So it would be like looking at your instruction manual and then you take note on the side and you use those notes to do, right, some action. That's exactly what happens here. Some transcription machinery, which we don't need to get into because it's a little bit beyond the scope of this, comes along, and what it does is it reads the DNA and it transcribes it, or that's the word for it, transcribes it. If that's confusing, all it's doing is it reads it and it changes it, makes a set of notes on something else, and then some action happens. That's as far as you need to think about this, right? And so if you think about this, what if I want to read this section of DNA over here? I can't, right? If the DNA is just like this, I can't read that. And so what that means is it's a dynamic genome. The reason it's dynamic is because it moves around. So as you can see over here, it moves around, right? This transcription machinery sometimes needs access to the stuff on the outside. Sometimes it needs this piece of DNA over here. And to do that, the genome has to constantly be moving around. And the reason for this, or the way that we know that it moves around, is because not everything in the DNA, not everything in the instruction manual is being done at the exact same time. And so that just proves to us that this genome is constantly changing in the sense that it's like moving around. So this is just another view of it. As you can see, it's a circle. And so sometimes this piece of DNA gets accessed. Sometimes it's this one. Sometimes you need this DNA in the inside area, right? So that's just to show you that the genome is dynamic and it's changing. Now, one thing of importance is that bacteria don't have membrane-bound organelles. So we talked about membranes in the last video. You can have a phospholipid bilayer or other kinds of membranes, but for bacteria and prokaryotes in general, they don't have membrane-bound organelles. So what do they have? So they have what we call bacterial structures or microcompartments. So let's start with one of the bacterial structures. This is called the thylakoid. It's a structure that enables photosynthesis, right? And the weird thing about this is it seems like it is a membrane, right? And in that sense, but it's not a membrane-bound organelle. The way it works is you have this membrane over here, the plasma membrane, right? This is the cell membrane. We're not distinguishing what kind of bacteria this is, right? But you have some sort of plasma membrane. And what happens is the thylakoid basically is a clever folding of the cell membrane, right? So it's not its own membrane. It's just using the cell membrane to create this kind of structure. So it's just a folding of something you already have. You're not forming a separate membrane. You're just using what you have in a clever way. And it looks kind of like this strange inner branching. Now, that's a structure, but there are also microcompartments. The difference is microcompartments are areas surrounded usually by a protein shell. And what they do is they allow certain reactions to happen. So let's get into some examples. This first one is the Carboxysome, and it appears as a giant dark spot over here. And so the function of it is to break down, it's what we call fixing CO2 gas, which is useless to the cell, right? The cell can't do anything with CO2 gas. And so it has an enzyme called rubisco that turns that useless thing, which is CO2 gas, into something useful, which is a carbon sugar, right? Now, let's break this down a little bit. Issue number one is that for this to function, it needs CO2 gas, right? But what happens is the cytoplasm that we're in, so let's go back. The cytoplasm, everything in here, is pretty much all water. So nearly 70-80% of it is water. And so what happens when you have CO2 and water? They will mix, and what you get is either H2CO3. You don't have to worry about the names. I'll just say it like this so I don't confuse you. You can have H2CO3. You can get HCO3 minus or CO3 2 minus. So this is basically just saying carbon dioxide plus water forms one of these three things. Now, the reason it's different, or it can be three things, has to do with the pH of the water. And the pH of a normal cell is about 7. So what you end up with is this right here, the HCO3 minus, right? You don't have to worry about the name, but just know that you end up with HCO3 minus, right? That's an issue right there, because we said it uses CO2 gas and it becomes a carbon sugar. So what we need is to solve that, right? what cells, prokaryotic cells have come up with is carbonic anhydrase. It's an enzyme, so a protein, that can take any of these three. So, for example, it can take the HCO minus, turn it into, it can take the CO3 2 minus, turn it into CO2, etc., right? So now we've solved that. We have our CO2 now, which is good because that's what we need over here. But that leads us to a second issue. Once we get the CO2 gas, there's still a bunch of oxygen usually in the environment, right? And so Rubisco, the enzyme that turns the CO2 gas into that useful carbon sugar, the issue there is Rubisco can either take in oxygen or carbon dioxide. So if oxygen binds, it'll just slow things down. It won't do anything important other than just slow our reaction down. So how do we solve that? What we do in the carboxysome is we form a protein shell. That protein shell is important. It's not a membrane, right? So I just want to specify, this is not a membrane. This is a protein shell. The reason we want a protein shell and not a membrane is because protein shells won't allow gases to diffuse in or diffuse out. As you can see over here, this is your protein shell. It won't let oxygen in and it won't let carbon dioxide out, which gives us two good things that we'll look at in the process over here. So let's start with our HCO3 minus, right? So this is not CO2. So what happens is it enters the cytoplasm as this, right? You can imagine CO2 out here enters and it becomes HCO3 minus because of the water that's in the cytoplasm. Now, when that happens, this will enter into the carboxysome. Now, the carboxysome takes this species, passes it through the carbonic anhydrase, and what that does is it creates CO2. So now you have your CO2 within the carboxysome. This is what I was talking about. It cannot do this. So this is not going to happen. Why can't it happen? Because of that protein shell keeping the CO2 in. That's the first pro of doing it this way. The other thing is oxygen can't get in, right? Meaning Rubisco, our enzyme over here, works just fine, really well actually. And then obviously it can do other reaction stuff, but that's the important part of the carboxysome. Now, another thing we have is the enterosomes. So the function of this is to take substance 1, which is useless to the cell, and turn it into substance 3, which is useful to the cell, right? And what happens is you'll form this intermediate right here, which we don't want. We don't want substance 2, but to get from substance 1 to substance 3, we do need to pass through this. And so that's our first issue, is that the cell needs substrate 3, but it only has substrate 1. So what can it do? What it does is it metabolizes or breaks down this first substrate into substrate 3. So that sounds great. You have substance 1 and you want to turn it into substance 3. Let's do it. Well, we ran into another issue. To metabolize or to break down substance 3, you need to make substance 2 first. And remember, substance 2 is deadly to the cell. See how I wrote toxic over here? So what do you need to do? Or what did prokaryotic cells do? They form another protein shell. This time it's called the enterosome. And it keeps the substance 2 inside and finishes the reaction. And then it lets out substance 3 into the cell. So let me show you what that looks like. Over here you have your cytoplasm on the outside. This stuff, right? Inside of the cytoplasm is this enterosome. So this is another protein shell. And what it does is you have your substance 1 that can enter the enterosome just fine. And then you have an enzyme that turns substance 1 into substance 2, which is toxic to the cell. So if this were to come out, the cell would die. So we don't want that. And the protein shell will not allow it to leave, which is exactly what we want. And then you bring in another enzyme into the enterosome, and that will create substance 3. And we end up with substance 3 in the cytoplasm, which is what we're looking for. So now that we understand that, there's also a few more, right? This one is a little bit simpler. It's just a gas vesicle, right? And it's kind of intuitive, unlike the other two's. Intuitive in name, unlike the other two. So this one appears white The function of it is literally to allow the cell to float or sink in a water column. This could be the ocean, this could be a pond, hydrothermal vent, whatever it is, as long as it's in water, it has this gas vesicle which can fill up, right? So when you're filled with air, you're going to float. When you have less air, you're going to sink. So that's what it says right here. More gas, you're going to float. Less gas, you're going to sink. So all you have to think about with the gas vesicle is it can either float or it can sink, up or down. That's it. Now, we have something called a magnetosome up next, right? These guys appear in a line, and they're made of magnetite. So if it's easy to remember for you, if you think of magnetite and magnetosome, all you have to think about is magnets, right? And it's pretty much what it is. So let's look at the function. You have a cell, and these purple lines indicate 3D space, right? You can move left, right, forward, back, etc., right? That was my attempt at visualizing that for you guys. So the first issue you run into is the cell needs nutrients or oxygen. It could need more nutrients or more oxygen, or it could need less oxygen and more nutrients. It really depends on what kind of cell it is. Not every cell wants a lot of oxygen. Just keep that in mind. So the solution is to develop a phospholipid membrane that makes magnetite. So you might be a little bit confused as to how these two relate. I'll break it down for you right now. Let's go back to this cell drawing. This cell is looking for nutrients and oxygen. So if this cell is in the middle of the ocean, for example, and it's looking for these things, it has to figure out where the nutrients are. So if it's wandering around for a long period of time without food, it's going to die. And so the way it counteracts that is to have this magnetite. What the magnetite does is it basically senses the Earth's magnetic field. And what that helps with is it allows it to go towards areas. So let me show you. Let's just say this is a magnet. Maybe that's not the best magnet. This is a magnet, right? Let's just say this is Earth's magnet and you're a cell. What happens is you'll be able to sense where that magnetic field is coming from, and you'll be able to move towards it to either get more or less oxygen instead of moving all the way around the ocean just to find nutrients. So this is just one way that is very similar to this gas vesicle, but instead of floating and sinking, this has to do with 3D movement in water. So what they do is they create this magnetite, and it follows the Earth's magnetic field, allows them to figure out where the nutrients or oxygen are, and it limits wandering in the water column unnecessarily. And we said before that there's usually no membrane, right? This is the only exception here where it does have a phospholipid membrane. The reason for that is because it has to make its own magnetite. And we won't go into exact excruciating detail on how this happens, but just know the phospholipid membrane, remember how it has selective permeability? So your phospholipid membrane allows ions in or keeps them out. This allows it to make magnetite at a very basic level. That's why you need a phospholipid membrane. But again, we won't go too in detail on that. Now, before we can get to the rest of the microcompartments that are possible in a cell, we have to understand what cells are made of. So let's take a look at what cells are made of first. They're made of macromolecules. So think carbon, nitrogen, phosphorus, and sulfur. If it helps you, you can think of CHONPS. I find this useful to think of CHONPS. What that does is it just tells you that it's carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And there's a little formula that you don't really have to memorize, but you can have it written down. It just says that the cell is C5H7O2N. What that's saying is basically the formulation or the kind of ratio of how many carbons, hydrogens, oxygens, nitrogens you need for a cell. It's basically just giving you sort of an understanding that these are the most important things that a cell needs and in what ratio to each other. Now, of course, there's also micronutrients, which we will not be going over because it's an entire table and it's very unlikely because it depends on the cell that you're looking at, and it's only in trace amounts, very little. So when you think of a cell, you're mainly thinking of these guys, not these guys. So now that we understand a little bit about what makes up a cell, now we can continue with the microcompartments. So there's something called a polybeta hydroxybutyrate, which is PHB. These appear white, and their function is to store extra carbon as a lipid polymer. You can think about this as a fat, right? So storing fat in your body instead of letting it out. So this is what a lipid polymer looks like. It's just a collection of lipids, right? A long chain of lipids. Now, the issue, as we see over here, and we went back to this idea of the CHONPS. So your carbon is really high, which is good, but you can't grow because your hydrogen, oxygen, nitrogen, phosphorus, and sulfur are too low for growth. So the solution, you don't want to throw away that carbon just because you're not growing. You store that extra carbon for later growth in these lipid polymers, which you can convert back to whatever you need once you have enough CHONPS to grow. Now, same thing with the phosphates. These also appear white, and their function is to store extra phosphorus in a phosphate chain. So what does that look like? This right here is your phosphate chain. So let's go back here. So in this case, things are different. Your carbon, hydrogen, oxygen, nitrogen, and sulfur are too low for you to grow, but your phosphorus is really high. So again, instead of throwing it away, you're going to store it as a polyphosphate chain. Poly just meaning many, and you can see that over here. You have a long chain of phosphate ions. Now, there's another one, the sulfur granules. This is slightly different. The reason for that is, first off, it appears dark, and you have to think about the differences between the sulfur granules and these guys over here, the polyphosphate granule and PBHB. These guys are limited in their growth, whereas the sulfur granules aren't really based off of growth. So the reason for this is because, first off, only some bacteria break down H2S into energy, right? And again, if you're not familiar with these terms, you can just follow along. So you have this H2S gas, right? Meaning sulfur. So this is a gas that's in the environment. Some bacteria can break that down and they use it for energy. This is their quote-unquote food source. Now what they do is they oxidize it, which you don't really have to know what that means. But all that's really happening is you have this H2S gas, and through this process of oxidizing it, you give away 2 electrons. And then you end up with this sulfur. Once you have your sulfur, you can give away 6 electrons when you oxidize it. And then you get to this last species, which is the SO4 2-, that can't give up any more electrons. And so what you see is that you've given up 8 electrons through this process, which is a lot of energy that can be, it's basically a lot of electrons that you can use as a form of energy. So the function of the sulfur granules is to store sulfur in this form right here, in this intermediate form, for later conversion into this SO4 2-, which is your sulfate, if you were curious what the name was. Now, this is not due to the growth limitation. This is actually just the bacteria preparing for a possible low H2S time. So basically, they're just rationing. So instead of using up your H2S, all of it, to get your sulfate over here, your SO4 2-, you just keep it as sulfur, right? Remember how sulfur gives away 6 electrons? It's a lot of energy, right? Versus over here, you've given up 2 electrons. So what they do is they ration this because H2S is a gas, and gases can disappear in a sense. Your environment cannot have it at some point. So you just want to ration it, not because you're trying to grow. Now, that's what I just talked about, is that issue of the H2S gas running low means there's no energy or food, and so they store the sulfur to ration it just in case. Now, we're going to move into something called sporulation, right? This is the highest form of protection that a bacterial cell can have. It forms what we call an endospore, and we'll talk about that in just a second. So the issue is, before we had issues with growth, right? We were either missing some phosphate, there's not enough to grow, and so we're collecting all this stuff, right? Now, worst case scenario, your carbon, your nitrogen, and your phosphorus are low. You're, in a sense, screwed, right? So what happens in that case? The only thing you can do is form an endospore. Now, keep in mind, forming an endospore is not for everyone. Only a small segment of gram-positive bacteria can even form an endospore. So let's look at normal bacterial cell division first to understand how endospores are made. So you have your cell over here, which is a normal cell. What happens is it'll replicate its DNA. So as you can see, it first elongates itself. As it's elongating, it's also replicating its DNA. And so what happens is, as it's getting divided, this whole process is called binary fission. And so what happens is your DNA is now one copy on the left, one copy on the right. Now what this looks like is, over here, you form what we call a constricting ring. So as you can see over here, the Z ring is splitting this thing down the middle so that you form two different cells. So this is the process of binary fission, which is normal cell division. Now this Z ring is important because it not only splits the two cells, so it constricts till these two divide entirely. At the same time, the FTCZ, the fifth Z, what it does is it also brings in the proteins. So remember back to our last lecture, we talked about the penicillin binding proteins. Those help to rebuild the cell wall of the bacteria. And so what that does is it both fixes this area right here and it splits the two in half. Now, this is normal bacterial cell division. The sporulation happens when we are missing a lot of our nutrients. So instead of this, we have our normal cell, and it basically, instead of going through normal binary fission, what happens is it splits but unevenly, right? There's not enough nutrients to go through a full division. And so what happens is you have this little area, which we call the forespore. Doesn't really matter the exact name, but you also have this huge mother cell. And so what it does is it basically engulfs it, right? It eats it, which is what we call phagocytosis. And then instead of going through that whole process of becoming two cells, now you're still one cell that ate the other one, and the mother cell will die, right? It'll basically explode, and you're left with the spore. Now, the spore has its own set of DNA, as you can see over here. It also has what we call dipyclinic acid. I apologize if I mispronounced that. This is going to become really important as well. But what I want you to notice is how the spore has so many layers. Just look at all these layers. 1, 2, 3, 4, 5, 6 different layers, right? Now, 6 layers usually means it's very protected, and that is the case. This, again, is the highest form of protection a bacterial cell can have. And so what you see is, again, it's only in a small segment of gram-positive bacteria, and the DNA is so heavily compacted that they've drawn it in this weird shape just to show you that it's not really loose like it was before. Now, the dipyclinic acid, which we'll call DPA, it also helps the spore survive even more. So what you see is that it allows it to survive extreme heat and harsh chemicals like hydrogen peroxide. So without the DPA, it actually survives less, even though overall this is the highest form of protection that a cell can have. Thank you for watching. I'll see you in the